for our Author Spotlight series at the Journal of Neuroscience Research. I'm Monica Santisteban, instructor in neuroscience at the Fowler Family Brain and Mind Research Institute at Weill Cornell Medicine. Today, we're highlighting an exciting paper published in JNR's September 2021 issue entitled Cleaved PINK1 Induces Neuronal Plasticity Through PKA-mediated BDNF Functional Regulation. We've invited the first author trainee, Dr. Smijin Soman, to talk to us about this paper. Dr. Soman is a postdoctoral research associate at the University of Nevada, Reno School of Medicine, with interests in understanding the pathophysiology of neurodegeneration and Parkinson's disease. Joining us today is also Dr. Soman's mentor and co-corresponding author, Dr. Ruben Dagda, who is associate professor, and his lab focuses on identifying the molecular mechanisms by which midbrain dopamine neurons degenerate in models of Parkinson's disease. So thank you both for joining us today.
Thank you.
Thank you very much. Thank you for the opportunity.
Yeah, so let's get right into it. So to start off, can you briefly summarize your findings and tell us what is the most exciting thing about your paper?
So the most exciting thing about the paper is that usually most of the field is looking into full-length PINK1 and its role in mitophagy. But our paper went beyond that, and it is actually looking into the post-translationally cleaved products of PINK1, the cleaved PINK1, and its potential role in neuronal development. So what we found is that increasing the expression of cleaved PINK1 can increase the levels of BDNF and in turn increase dendrite length and synaptic plasticity. So this is one of the major contribution from this particular paper.
Thank you. That's so exciting and such a different way to look at PINK1. So can you add on to that and tell us what is something that surprised you about your project, or maybe what was the most unexpected finding?
Yeah, so one of the most exciting data for me was that we transfected cleaved PINK1, or we overexpressed cleaved PINK1 in primary cortical neurons, and we saw increased levels of BDNF. So this was the primary data. But apart from that, we also observed that the cells in the proximity of the positively transfected, cleaved PINK1 positively transfected cells, they were also showing signs of the phenotype, which is like increased dendrite length and such. So then we were thinking that maybe there is not just an increase in expression of BDNF, but there's also an increase in the release of BDNF into the media, and that BDNF acts on the adjacent cells. So we checked we checked the levels of extracellular BDNF, which is in the media, through ELISA test, and we found that there is also increase in release of BDNF after overexpression of cleaved PINK1. So this was one of the very exciting results for me.
Yeah, that's really cool. I actually, now that we're talking about, like the PINK1 effects, I have a question about the activator that you're using.
Yes, sure.
Yeah, is this activator specific for PINK1, or does it have maybe some off-target effects as well?
So kinetin is a pharmacological activator of PINK1. So what happens is that kinetin metabolizes inside the cells into a KTP, kinetin triphosphate. So this is an analog of ATP, and the PINK1 has a binding site for ATP, and ATP activates PINK1. So KTP acts as an analog of ATP there. So it is. There are multiple studies which say that it is very specific for PINK1 activity, increasing PINK1 activity.
Okay, great, thank you. So to add on to that, so what studies are you working on now or planning to do in the future?
So in this particular, in the published paper itself, we have shown data that the cytosolic PINK1 increases BDNF levels through PKA. So PKA is the intermediate, so cleaved PINK1 acts on PKA, and PKA causes increase of Krebs phosphorylation, and Krebs in turn increases BDNF levels. So this whole PINK1 PKA Krebs BDNF signaling is what causes increased dendritic plasticity. Right now what I'm working is on to figure out the spatial temporal dynamics of this PKA activation by PINK1 through FRET assay.
Okay.
So this is one of the studies which I am looking into right now, which is very closely related to the paper which we have published in JNR.
Yeah, that's exciting. Hopefully whenever you have, your next work, you'll also come back to JNR and publish your findings.
Sure.
So, Smijin, can you tell us a little bit about yourself and your career goals?
Yeah, sure. So I did my PhD from India, from Cochin. That was back in 2013. Then I moved to Europe. It was a European Union collaborative project. So partly I worked in UK, in Sheffield, and partly I worked in Warsaw, Poland. I was working in the same protein, PINK1 itself, but it was a zebrafish model. It was a PINK1 knockout zebrafish model, and I was looking into role of PINK1 in mitochondrial calcium homeostasis. So from there on, I wanted to continue my work in PINK1 and neurodegeneration and Parkinson's disease. So I found a great mentor, Dr. Ruben Dagda, here in University of Nevada, Reno. So I moved here back in 2018, and I'm working on the project since then.
That's great. Awesome.
Yeah.
And then I guess maybe just last interesting question. Do you have any thoughts on the neuroprotective or potentially therapeutic implications of PINK1 activators to increase BDNF levels?
Yes, actually, it's a very important question. One of the challenges we also faced is that kinetin needs to be metabolized inside the cell, so the dynamics is pretty long term. So that particular dynamics has to be standardized to look into more a therapeutic version of this particular activator. But there are certain people who have been looking into this, and there is actually a company working on this one.
Okay. Yeah, that's so interesting.
Thank you.
Okay, so we wish you the best of luck and are definitely looking forward to your future work. We really appreciate you joining us today to discuss your paper. Thank you.
Thank you very much.